Hello, everyone. I am so happy to welcome you all to the second webinar in our series about keys to a successful natural history study. My name is Kathy Zhang, and I am the Associate Director of Project Management and Clinical Innovations at PROMETRIKA. We are a full-service CRO located in Cambridge, Massachusetts, and we've had the good fortune of working with many of our sponsor partners on natural history study development, especially in rare disease indications. I will be your moderator for this discussion, and I am joined by a special guest, Frank Stout, who is the Director of Clinical Operations and Head of Early Clinical Development Operations at Apellis Pharmaceuticals. We also have Chelsea Ryan, PROMETRIKA's Associate Director of Clinical Operations and Pharmacovigilance, as well as Kathy Holt, PROMETRIKA's Director of Data Management. I'm very much looking forward to spending the next hour with this panel of experts, as well as with our audience, to hopefully share our collective experiences around this very relevant topic. So natural history studies are starting to gain more and more interest within our industry. People used to think of them as simply less robust clinical trials that academics ran. You simply didn't have to try as hard because the same rigor didn't apply for natural history studies, as was commonly held. But now more and more natural history studies are starting to become a critical and even necessary part of a sponsor's development plan, especially for rare disease indications. And the FDA is now even recommending them in many instances. And as we all know that when the FDA makes a recommendation, it's more of a do it or risk our wrath. Consequently, we don't have the same luxury of treating these natural history studies like a bonus data source any longer. They have to be executed with the same level of planning and forethought as you would a randomized clinical trial. And I think this is where a lot of people get themselves into trouble, where they think that the same principles of running a randomized clinical trial are identical to those of a natural history study. And then they just go on autopilot. They apply the same monitoring principles, the same database design principles, and the same analytics plans. Thankfully, our extensive experience working with our sponsor partners on designing and conducting natural history studies has taught us better. It's taught us, the need to stop and think about what makes, for example, a prospective natural history study different from a retrospective natural history study, and how your strategies across all the functional areas need to adapt accordingly. And so this need for adaptation also highlights the need for highly experienced team members to work on natural history studies in particular, because the autopilot just doesn't work for studies when you're diving into the great unknown. You need that flexibility to be able to think outside the box for these types of studies. So in our previous webinar, we covered these themes as well as provided a case study for conducting natural history trials during COVID-19. During that time, natural history studies across the board were deprioritized. And so sponsors and researchers needed to come up with more creative ways of getting that natural history data. And so fast forward to, hopefully post-COVID times, those same lessons apply to all natural history trials. And so in today's webinar, we plan to take the discussion one step farther by presenting some practical, operational, and data collection challenges. We plan to discuss how to overcome operational challenges of executing natural history studies for both rare and non-rare diseases. We'll discover the role that technology can play in natural history studies, and we'll discuss how to design a database that is fit for purpose for a natural history study, instead of trying to fit a natural history study into a database design that's really more optimized for randomized clinical trials. And so with that, I am happy to introduce Chelsea Ryan, who will be talking about the practical operational challenges.
Thank you so much, Kathy, and thank you so much to all of our webinar attendees for joining us today. In the coming slides, I'll be presenting along with my friend and former colleague, Frank Stout, on practical operational challenges and recommendations for strategies to overcome them during execution of a project. In our first webinar series, I reviewed 4 common pitfalls with natural history studies: losing patient and investigator interest, overlooking regulations and guidelines, failure to consider real-world behaviors, and undervaluing the use of technology and study vendors. Today, we'll be providing recommendations on best practices as well as examples that Apellis has employed to build in quality by design in their execution of these types of studies. The first thing I'd like to talk about is building a clinical monitoring plan with quality by design. You're really thinking about creating your monitoring plan by considering the long-term goals and strategic plans your company has for the data. Plan to review your monitoring plan throughout the course of the natural history study to ensure that the plan grows with the goals that your company has for the use of that data. And then progressively elaborating your natural history goals. One of the things that we really learned about is that your natural history study can evolve over time. Especially in rare disease, you start a natural history study with very little information available to you, and you're using this as a source to learn more about these patients, learn more about what's important to them, and then build for future studies potential endpoints, build potential data that would be important to have for this patient population. And so you need to really think about ways to progressively elaborate those goals by involving your stakeholders throughout the process, having literature searches built in, and also go back and review your pipeline. As a sponsor, your goals as a sponsor company will change over time. Your pipeline will change, what you want to do will evolve, and you really should come back to your natural history study and use it as a tool for you to support your pipeline goals as a whole. We recommend focusing on the following areas to ensure your natural history study is kept fit for purpose, so that it does not become an overwhelming data set that's difficult to manage and monitor. So the first one is engage the patients, engage expert stakeholders, and a risk-based monitoring approach. Let's dive into that a little bit deeper. This is an example of a timeline for building in progressive elaborations of your natural history study into the execution of your project. I'd like to point out a couple of things. So at the beginning of your project, before your database is built, you're engaging your stakeholders. It's really important to engage those expert stakeholders, engage the patients in the conversation as early as possible. This is especially important for rare disease. You want to make sure that your future studies are studying things that are important to the patients, that will have a meaningful impact on their lives, and really make an improvement on their day-to-day life. So engaging that patient population early, engaging your stakeholders, and have them involved in your protocol development stage, your project planning, project kickoff, really crucial to the process. And Frank will talk a little bit more about this soon, about how Apellis has engaged expert stakeholders in their natural history study development. The next thing is then you would, for the blue, these are database elaborations. And what this is saying is you have your initial database release, and then you have planned time points to go back and learn more about what your goals are, how they could have changed, how new information that's come to light since the original release of your database for your natural history study and the design of your protocol could inform you to make changes that could make your natural history study data set more impactful for the future. So this is just an example, but it's over the course of 4 years. It's saying, you could engage your steering committee meetings, schedule your steering committee meetings with your key opinion leaders twice a year, have patient engagement milestones every single quarter, and then making sure that you're doing a literature search at least once a year to see if there is any new published literature that could inform your decisions for your study. And then again, you're building in those learnings. I think the big takeaway here is you don't know everything when you start a natural history study. You rarely do. So you need to build your plan for the natural history study with that in mind and build in continuous learning cycles, and then take those learnings and put them back into your study and change your monitoring approach based on what you're learning. For this slide, I'd like to talk about engaging expert stakeholders. So the key here is that, again, you engage your stakeholders early and then engage them often. You need to build in time points throughout your study to pull your stakeholders together. Some examples that we have are your key opinion leaders and steering committee meetings. So I gave that example of maybe making 2 time points throughout the year that you have prefixed meetings with those groups. Investigator meetings. So in interventional trials, we often will have, weekly, bi-weekly, monthly investigator meetings where you're pulling all your investigators together and you get feedback from them about how the study's going, how recruitment's going, what some of the barriers are, what they're experiencing. This should be treated no different. You really should talk to your investigators and see if they have any feedback directly from the patients and then also from the investigator experience. Partnering with patient advocacy groups, and Frank will talk a bit more about this, but this is definitely key. And then again, regular review of the literature. So I'd like to hand it over to Frank. He's going to talk a little bit about stakeholder integration. One of the key things here is, sponsors and CROs in our relationships, we pull together the right group of stakeholders, and the sponsor will bring some stakeholders to the table, and then the CRO can really fill in those gaps to ensure that there's a holistic approach to the stakeholder group that is working on a natural history study. So I will hand it over to Frank to talk about Apellis's approach to stakeholder integration.
Thank you, Chelsea, so much for introducing this part. This is very critical when we're looking at developing natural history studies. As many of us look within sponsors and CROs, we have many functions, and we all might call them something a little bit different, but we listed a few of them on the slide here that are critical when looking at developing these studies. And so we have typical clinical operations in your medical, your MDs, your patient advocacy groups, your health outcome groups, and your commercial groups. It's critical to also keep these in mind. You might not always think about these 2 functions within your company when developing the study, but they also can look at your quality of life questionnaires, which are also very critical. And biostats and data management, these of course are always utilized within our clinical trials. But one key piece that we will talk about later too is data management and making sure we do a fit for purpose on the EDC. It isn't that we always have to use the same EDC for every single study. So making sure you actually understand and develop what you're looking for. And the other 2 groups is your preclinical and your regulatory group. Another key stakeholder with developing these studies are your biomarker specialists and your technology vendors. So many of us, we can use within the sponsor, the CRO, and our KOLs, a biomarker specialist. And we do this at Apellis all the time. We look at people across the whole entire world within academic universities and or laboratories. And we find people who have been focusing in areas specific to what we are trying to develop. And we all know that the FDA has been pushing this, and we have been utilizing many vendors on the technology side, whether it's Fitbits, whether it is virtual reality goggles for ophthalmology, whether it is iPads. One other group that Chelsea has talked about as well is the patient advocacy group. We can't forget about patient advocacy groups and the patients, because this is a key portal for how patients get back to the sponsors and relay what is difficult for patients and what they want to focus on. And then last but not least, of course, is our key opinion leaders and our PIs. Bringing them on early, as Chelsea also said, is critical in developing a complete package. So going forward, Apellis has looked at developing steering committees. And I've said this many times, but engage early. And I say this because when you're doing this, you're really engaging to find those champions. You find them before the final protocol. It helps you develop the study and also helps you find physicians across the whole entire world, if your study is global, that can be your champion, to get the patients in the study and also keep them in the study. Another key piece that we don't do all the time, and we do within our interventional studies because of safety monitoring committees, is review the data. Reviewing the data helps you pivot and make sure that you collect all the data you need, so at the end of the study, you can actually make informed decisions. And then one of the last pieces too is make sure you review the data before you start your study. You need to make sure that you build and help build your interventional programs. Another area that we've looked at to help develop our pipelines is also look at your off-target endpoints. Make sure that they support your total strategic goals. And one piece I continue to bring up always is patient advocacy groups. Make sure that you understand the patient hurdles on everyday demand so that your schedule of assessments when you're developing these studies is actually feasible. Thank you. And that's all I have.
Next, I'd like to talk a little bit about risk-based monitoring. I think that this can often become a confusing topic for people when it comes to natural history because you're not really sure how much you really need to monitor this natural history data. You don't want to over-monitor it, but you also need to keep in mind the future use of this data could likely go in for an FDA submission as a historical control. And if that's the case, you do need to approach this data and the monitoring of it with the same rigor that you would a regular interventional trial. One strategy that we have is looking at the percentage of the SDV and thinking about the data points that you're collecting in your natural history study, which ones will be future endpoints, potentially future endpoints, or contributing to the decisions of future endpoints. Any critical safety data that you could be using as maybe a baseline to understand the population and what would be the usual prevalence of an adverse event for that population, you would want to SDV that data. Any natural comorbidity data, where you're trying to build out patient profiles, especially in rare disease, if you don't know much about the population, anything that would help inform future investigators' brochures or decisions about how you'll design your future studies, you would want to SDV that to a certain degree. One thing that we will talk about is trying to consider ways to reduce your clinical monitoring costs on natural history studies. These studies do tend to be long and they can be costly if you don't think about that up front. And technology can come in useful here with direct-to-data capture, centralized data analytic tools, ePro and wearables. Frank spoke to a few, and Kathy will be talking more about this shortly. You can reduce your clinical monitoring needs. You don't need to SDV anything that's coming directly from the patient into the EDC or from the EMR directly into the EDC. So that can help. And then also trying to work with sites that support remote monitoring. This can be a difficult one, especially if you're working with rare disease and your options for the sites that you work with is really driven by where the patient population is. But just something to keep in mind if it is an option to activate sites that do support remote monitoring so that you can reduce your travel costs for the natural history study. And talking about informed consent forms and GDPR, this goes back to making sure that you're treating a natural history study with the same rigor that you would an interventional study. You do need to have every patient in the trial sign a consent. It needs to be an IRB approved consent. If you're working with any patients that are in the EU, you would need to follow GDPR guidelines. One area that I'll just touch upon here is your risk-benefit analysis section of your ICF will be slightly different for this type of a trial. You're not giving them an intervention. You're not enrolling them with an intention to treat. So the benefit to the patient is not immediate. And it's important that the doctor has that conversation with these patients that oftentimes, they're contributing to the knowledge base that will hopefully make future treatments available to them through the future trials that this data will support. And the risk is minimal. So that's a conversation and some language in your ICF that will be slightly different than what you would see in a typical trial. And then again, you're designing with the end in mind. So continually going back and looking at the goals of your study, the goals of your research, what you've learned, and incorporating that into your risk-based approach. You may learn that, like Frank said, that there's an exploratory biomarker that all of a sudden becomes important to this patient population. That may be something that, in a future iteration of your monitoring plan, now you want to monitor. This slide just tells you that monitoring oversight responsibilities of a natural history study really require the same monitoring activities as an RCT, with very few exceptions. There's no drug supply and there's no safety management. So when you're going to build out your monitoring plan and you're trying to create potentially a template for your company, I would advise to try to keep in as much as you would usually do on an interventional trial, and then just strip out the drug supply, safety management, and have some type of a risk-based monitoring approach on all the other areas that you would typically monitor. This is a quick example, and it starts off like any other trial with a trial risk assessment meeting. We would advise that you do that annually to meet ICH GCP guidelines and have reassessments to see if your protocol amendments or what's going on in the industry has changed the risk in any way. And that risk is not necessarily the risk to the patient because there's no drug involved, but you're talking about like the risk to your data collection, the risk to you meeting your goals as a sponsor. So looking to make sure that you're monitoring the data in a way that avoids any risk from occurring and becoming issues down the line. We would recommend that you do an on-site either qualification visit or a site initiation visit to make sure that these sites and the facilities that they have meet the needs of the study and any future study that you would have, that they would meet the same rigor that you would qualify a site for a future interventional study. For remote interim monitoring, this is just an example of where you could do every 6 to 8 weeks, depending on the data collection points and the frequency that you're trying to get data from these patients. And then you would do a targeted STV, talking again about those fields that are most important to the long-term use of the data. This can all be done remotely if the site supports that. And that's how I would recommend it if you can do that with the sites that you have on your natural history study. And then the ICF review, you still would want to do some level of ICF review to make sure that these sites are following the ICF consenting process correctly, abiding by ICH GCP. One example is that you could do maybe 100% of the ICF review of the initial 25% of the patients, and then you could do 50% thereafter. And that could be flexible depending on the risk, depending on your relationship with the sites. You're really just looking to make sure that the site has a process in place and that they are correctly ICFing or consenting to patients. The last piece of the puzzle is engaging the patients. Frank's going to talk a bit more about the ways that Apellis does this, but some recommendations and strategies that we've seen work well are newsletters, reaching out to patient advocacy groups, reaching out to patients through those advocacy groups, reaching out to patients through social media, and then patient study satisfaction surveys. So at those quarterly time points where you're touching base with the patient, in the example that I gave with the timeline, an example could be, maybe once a quarter, a year, you survey the patient to see if the data that we're collecting, if there's anything that's important to them that maybe we're not collecting. You could ask them about their daily life. You could ask them about the amount of work that they've had to do to participate in the study, if it's been a burden on them. Any questions like that would be great feedback to have from the patient to understand if you can design the execution of your project in a way that is a better fit with them and their life to get a better data set that's easier for the site to collect and the patient to provide. So I will let Frank give an example of how Apellis does this. They are a great sponsor in engaging their patients and making sure that their approach is very patient-centric.
Great. Thank you so much, Chelsea. So we are very keen on making sure we engage with our patients early and often. And when we look at this for natural history studies and rare diseases, it's super critical. And we always kind of look at this sometimes only one way, and it's actually a two-way street. So it is just as beneficial for us to engage with patient advocacy groups on the front end to understand the risks, understand the patient burden, to help us build the study, but it's also super key because the patient advocacy groups, as Chelsea noted, can help us get the awareness out through newsletters, social media groups, and websites. And one way we have done this is we have gone across the world, and I can specifically think of a time that we went to the Asia Pac region, and we spoke at a town hall in conjunction with other physicians where patients were able to ask questions, but we were also able to ask questions for patients to understand the route of administration. Is there a difference between cultures and different areas of the world of how patients perceive your route of administration, which is critical when you start looking at how you're going to develop your pipeline and your clinical development plan. In addition, we have also been very keen on making sure that we bring forward our natural history studies because they aren't the easiest studies, as we all know, to enroll, and they play a key part in rare diseases. So once again, I can't stress this enough, making sure patient advocacy groups know about your study through social media groups. There's tons of groups on Facebook now. There's tons of groups on Instagram. They have their own websites. They have their own nonprofit groups now. It is key making sure that they know about you just as much as you're trying to know about them. And one last piece that we've actually started doing a few years ago, and something that we don't always think about when we're looking at doing our press releases out on the wire, is making sure that we actually change the wording for patient advocacy groups. So I know within Apellis, we look at making sure we make it patient focused when we talk to those patient advocacy groups so that when the patients read it, they know what we're doing and they can follow us. Like ensuring the patient knows how you are progressing within their disease is critical to making them a partner on your journey. Back to you, Chelsea.
Thank you so much, Frank. To summarize our takeaway messages from the clinical operations portion of this presentation, we really want to emphasize the importance of engaging a diverse set of stakeholders, including the patients, as early as possible, and then also throughout your execution. Developing a fit-for-purpose RBM approach, and then progressively elaborating your goals. So making this a constant cyclical learning objective for your company, where you're going back and you're continuously learning about this patient group that you're studying, their needs and the goals for your company and how they align. Regular reassessments of your data management methodologies, and then leveraging technology to help make that process easier. With that, I will hand it over to Kathy Zhang, and she'll talk to you about a few of the technology options out there that we have seen work well for natural history studies.
So just because natural history studies are becoming more necessary, it doesn't mean that the data has become any easier to collect. And as you all know, in many ways, the data is actually harder to collect in a natural history study than in a randomized clinical trial. And so sponsors have started to lean into technology to help facilitate the execution, especially for rare disease therapeutic areas. So while most of this technology has been around for a while, sponsors are often reluctant to use them for various reasons, some of which is business or cost-driven, some of which is lack of experience. So running a rare disease natural history study actually forces them to explore technology alternatives to traditional data collection methods, because all the normal problems of a clinical trial are exacerbated in a natural history study. So for example, data quality issues, data quantity issues or lack thereof, subject retention issues, and so on and so forth. The small sample sizes, especially for rare disease natural history studies, mean that in order to cast a wide enough net to capture any and all meaningful data, you can employ technologies to help, such as e-consent, e-pros, wearable devices, etc. So you might ask, well, how is this different from running a randomized clinical trial for a rare disease? Well, the primary difference is that you're diving into the unknown with this therapeutic area when you're running a natural history study. So instead of knowing a priori that you want to collect these endpoints at these time points for these primary outcomes, it's almost like an Easter egg hunt where your goal is to try to collect as many eggs as possible, just in case they might prove useful in your clinical development plan at some point down the line. And data analytics can really be useful to help identify trends amongst all the data that you've now collected on a continuous basis to help with that progressive elaboration of your natural history study goals, as Chelsea had mentioned. So these analytics tools, like the ones that PROMETRIKA uses, can produce visualizations like graphical patient profiles, which helps you visualize the data on a per patient level with live data at any given point in time. So one interesting side effect that came out of having to run natural history studies during COVID was that it acted like a catalyst for the development of innovative data collection methods. Sponsors turned to these commercially available direct-to-patient data sharing platforms where patients actually grant consent to provide medical data prospectively and access to their medical records to obtain data retrospectively, all for research. And so sponsors can use this data to build a natural history data set, or they can use it as a control arm for future studies. We've also seen that the data from these types of platforms represents just one source out of many that sponsors are using to build their natural history data set. And this idea that you can potentially have many different sources of data, where some of the data is collected prospectively while others are collected retrospectively, it can really represent a problem for the data collection and cleaning if you originally planned to run the study just like a regular randomized clinical trial and built your database as such. And so with that, I'm happy to introduce Kathy Holt, who will be talking about the data collection challenges of a natural history study.
Thank you, Kathy, and welcome everyone. In these next couple of slides, I'm going to discuss CRF design and database challenges and considerations for a natural history study. Natural history standard and disease-specific forms will differ from your randomized clinical trial forms. Critical forms like the demographics, disease progression forms, labs, vital signs, and concomitant medications, which are usually fairly standard for your randomized clinical trials, will likely need to look different from natural history studies in order to collect ancillary data. For example, on the DEMOG form, you may add questions about a subject's education or country of origin, nationality. On the medical history form, you may need to include questions about smoking status, specialty of the healthcare provider responsible for the clinical management of the subject at the time of symptom onset and diagnosis, or clinical manifestations at the time of diagnosis, requesting for very specific clinical manifestations, and family history, number of siblings with the disease. Most importantly for the CRF design is that the time frame for the collection of data needs to be considered and assessed in relation to data being collected. For example, an example would be to only collect data for a certain time frame, like 6 months from a diagnosis or a hospitalization or a significant event. So along with your CRF design, you'll be creating a very detailed and specific CCGs, even beyond what you would normally specify for a randomized clinical trial. For a retrospective study, the CCGs will be very specific about how to collect the data that will include specific guidelines around timeframes for the collection of data, how to handle missing data or data that is not available in a chart. The CCG should also include where to collect that data from, like for laboratory tests or imaging tests. The CCGs will also include how to handle data from multiple different sources of data. There are a few significant database build considerations to keep in mind. The database should be simple and flexible for when you have to make changes to the database following progressive elaboration that Chelsea described. You may need to redefine the collection of data to include more or less data. Other database items to consider are the visit structures. They can be simple, less dynamically added forms and folders, or no visit structures for retrospective studies where you would only be collecting forms. SDV requirement is an important consideration because you need to know what data will be source verified or not. So working with the clinical team during this part of the database build is important. SDV requirements will also impact data cleaning, as the data being entered may have more errors if not being source verified before entry. These SDV requirements should be specified a priori to avoid any database locking issues. This next slide represents a database strategy that we created for a natural history study that we were going to build 3 studies into one database that included 2 prospective studies, a registry and a prospective natural history, and a retrospective chart review. As you can see, there were some overlapping visits between the 2 prospective studies that we needed to consider. We determined that the retrospective chart review study could be included into the one database by having the site manually add forms because the data was being collected directly from the subject's medical record. The subjects in that second study would roll over into the chart review study. There were many database build considerations that we needed to consider for a one database design approach. These are some of the major build considerations. The subject ID needed to be robust since being used for 3 studies, and the default matrix would need to be clearly defined. Does it include visits and folders and forms, or is it blank to allow for the data to come in for these 3 studies? The data output could be problematic if you only wanted to see the data for just one of the studies. So we thought about creating a hidden field on all the forms so you could review the data for one study at a time by using this hidden field to sort the data. Locking the databases would be a challenge, and that workflow would need to be clearly defined with the clinical and stats teams. Post-production changes could be tricky to handle depending upon the types of production changes, since we are effectively running 3 different studies in one database. The challenges to a successful build were greater with the one database strategy. So before, during, and after the entry of subjects in the database, the DM must make sure that the data cleaning process is defined in the data management documents like your DVS and your data management plan and your manual review guidelines. The DM will need to define how to handle data discrepancies, like a missing or illogical data, which will be very important for cleaning of a retrospective studies, since issuing a query may not be feasible. The query process should not be impacted for prospective studies. If during review you are seeing too many data issues, then you may need to redefine the collection of data and let the clinical team know and the stats team know so that you all can work together to resolve those issues. Again, as stated, DM will need to work closely with biostats to define rules around how to handle missing and illogical data, especially for the retrospective studies. So in closing, as stated, data may be coming from multiple sources, which will complicate data collection and cleaning. But with thoughtful understanding about database design and build and cleaning, your natural history database can deliver the data that you would like to see.
So many thanks to Frank, Chelsea, and Kathy for all the great discussion points. I think the overall take-home message is that you can really maximize the utility of your natural history trial so long as you go into it understanding that the principles are not the same as those of a randomized clinical trial. And you have to plan for those differences accordingly. In the 3rd chapter of our webinar series coming in the winter of 2021, we plan to examine natural history studies from a statistical perspective. Similarly, as we've discussed here, one of the key mistakes that is often made is treating the statistical analyses of a natural history study as you would a regular randomized clinical trial. And by doing so, you actually risk losing a lot of the value of your natural history study by doing so. We look forward to seeing you all there. In the meantime, we would like to now open the floor to our attendees to ask any questions you may have about what we've discussed today. So please send your questions via the chat function of the webinar. So one question it looks like we've received so far from the audience is, what does PROMETRIKA use for analytics tools? And that's a really good question. It's really been a journey to look for an analytics tool that's really fit for purpose. Our search began with a series of requests from multiple sponsor partners to look for a tool that could really support descriptive analytics and not the advanced analytics that a lot of traditional analytics products can produce. So analytics tools, as most of us understand, originated, or rather their use in clinical trials, started with their use in wanting to analyze big data issues. So genomics data, even natural history data, and then our sponsor partners came to us and said, well, what if we needed a platform that ingested smaller quantities of data? And we didn't need a fancy, AI capabilities that a lot of these advanced analytics products produced. What if we wanted something that our medical monitors can just go in and on an ongoing basis run analytics with? And so PROMETRIKA, we partner with a company called SAMA, formerly known as Comprehend. And through a fairly exhaustive search, that is the product that we ultimately landed on for that purpose. And so, as we were talking, several questions popped into my mind around what Chelsea and Frank and Kathy Holt mentioned. And one question I had for the team actually is, we talk about the need for the planning activities of a natural history study to be nearly as robust as that of a randomized clinical trial. However, I do acknowledge and understand that one of the pain points of running a natural history study is that, there's often so much that is unknown about the therapeutic indication that sponsors are just chomping at the bit to get started, right? And so my question to Chelsea and Frank is, in your opinion, is it better to just get going with a natural history study, just so you can start that data collection and then elaborate as you say, to refine the process as you go? Or would you recommend that, our sponsors wait until they have a fully baked plan before initiating any activity?
Sure, I can take that question. Thanks so much, Kathy. I think that, with anything else in clinical trials, it's you need to approach it not so much as a cookie cutter, like you do this or you don't do this, but you really think about the study, the indication, what your goals are as a company, and then make a decision from there. So my response would be, I talked a bit about how for natural history studies, you still need to do a trial risk assessment meeting where you sit down and you're looking at the risk to collecting your data and the risk to getting what you ultimately want to help inform your future studies. And I would pose that question that you asked in that way and really look to see, if you started your study with what you know today, is that going to put you at risk for the future? And if the answer to that question is no, it's just going to make you ahead of the game and you can elaborate your system, that you're going to have those built-in time points that you would update your EDC, then I don't think that there's a real risk in doing something like that. It's just going to, allow you to start that learning process early. And if the answer to the question is, okay, we've gone through these questions in our risk analysis, and we really don't have enough information to build a meaningful database at this point, then I wouldn't invest your resource, time, and money in rushing to build a database. I would instead put your time and your money into trying to investigate what would make sense for you as a company to put into your natural history study, and then move on to create your EDC. Thanks.
I couldn't agree more with Chelsea. I mean, it really is a risk-based approach here, and you don't know everything when you start a study, especially a natural history study, and definitely for rare diseases. But in addition, you don't want to make your natural history study become too much. So I think focusing back on having the discussions with your KOLs, having the cross-functional discussion and understanding what you want to put in your natural history study to initiate it, and then make the analysis. Okay, will this give us what we want? Or do we not have enough information and we need to sit back and go find that information? So I think both ways you can look at this, but definitely it's definitely a risk-based approach.
Perfect. Thank you so much. So thanks to everyone who's submitting questions. We really like to see these, so keep sending them. So the next question we've received is, what are some of the unique considerations you think need to be taken into account for including digital endpoints into a natural history study, particularly if these endpoints haven't been used before? And that's a really great question, because I think a lot of our sponsors, a lot of our sponsor partners are asking themselves this question. And I think what makes this a unique challenge for natural history studies, and it may be stating the obvious, is that, as I mentioned previously, it really is like an Easter egg hunt. And your goal is to try to gather as many of those eggs as you can, just in case they might be useful in the future. And so for a natural history study, I think typically what we see and what we recommend is that really is your opportunity to pilot the technology that you're considering to use in your later phase studies. So if you've never used this piece of technology to look at this particular endpoint before, I think where some of our partners get into trouble is they think, I don't want to spend this upfront initial investment on the technology because it's too expensive, because we don't know enough about it. Let's just wait. Let's wait until it the payoff is greater, let's say. And so one of the mistakes is that they end up starting their journey into technology, if you will, too late into their development process. And at that point, you discover all sorts of clinical impediments to this specific piece of technology. So for example, you realize that this rare disease population has, let's say, tactile sensitivities that you weren't aware of. And so, you can't put a wearable around their wrist. Or let's say it's sort of analytics challenges with your vendor. So you might run into issues where the epoch length of the data collected is not short enough to capture tremors, for example. And if you didn't do that pilot with that vendor, with that device in that therapeutic area during your natural history study or during your earlier phase trials, by the time you realize that you have those issues in your later phase trials, it may be too late. So that's a really great question. So Chelsea, so you talk about the need for the progressive elaboration of goals throughout the conduct of natural history studies. Does this elaboration process involve conferring with the regulatory authorities as well? Are they willing to engage in those discussions during the conduct of, as early as the conduct of a natural history study?
Definitely. I think that, the FDA has quite a bit of focus right now on rare disease as well as patient centricity. And I think natural history studies, Frank talked a lot about this in his slides about incorporating the patient's voice, working with patient advocacy groups. Your natural history study is your opportunity to make sure that you're creating endpoints that are meaningful to your patients in the end. And the FDA is more than willing, and my experience and what I know of what I've read, to have those conversations during a natural history study, to make sure that you are setting yourselves up to create meaningful studies with meaningful designs for the patients in the end.
Great, thank you so much. So we have a few really great questions about the database. So one is, Kathy, you mentioned using one database instead of three databases for natural history studies. However, this posed the challenges that you described. Has PROMETRIKA experienced using multiple databases to capture the natural history data, and is there a benefit to this approach?
So we went through the route of just using a database for each natural history study instead of capturing them all in one database. So it was a benefit to use a single database for each natural history, and then to combine any data needed on the back end at the back end, instead of having it all in the one database after all of what we went through and diagramming out that three studies into the one database.
Yeah. And just to add to that, I think it speaks to this idea of, best laid plans where for economic reasons, you may want to, consolidate all the data into one database so that the data structure is all the same. But I agree, Kathy, that we have seen where you almost try to build too much into your one database, and the downstream considerations for, what do you want to do if you just want to lock one component? What do you want to do when you just want to do a data extract out of one component? I think practically speaking, as we've seen from our experience, it is ultimately more straightforward to have a separate database for each.
Yeah,
That's great.
And
Yeah.
I think it's also helpful from a clinical point of view in controlling the data and monitoring the data, to take that approach as well. It goes back to what Frank was saying about making sure that, you're not just throwing everything in there just to throw everything in there, and then it becomes uncontrollable and it becomes, difficult to manage as a sponsor. You want to make sure that you're creating some type of structure and form to help you be organized in your methods.
Yeah. And so another question around the database design, Kathy, is if you have an ongoing natural history study and you want to add or modify outcome measures, how would you do this from an operations perspective as well as from a database perspective? So, so both Chelsea and Kathy, I think it goes back to what Chelsea was explaining about how you sort of have to progressively elaborate. And so do you want to provide more details around that, Chelsea? And then perhaps Kathy can add to how you would do that with the database.
Sure. So if you wanted to add more, I think one thing you need to consider as you elaborate your database is I would caution taking things away, right? So if you're just looking to add, and as you learn more, then you add more information in there, you add more outcome measures, then you need to think about, just like you would with an interventional database, if you're going to go back. So are you going to go back on the patients that had already entered data into the database and try to retrieve that data retrospectively from them if you're working on a prospective? I think you would need to think about, what if you do a retrospective. I would take a risk-based approach to make sure that you're monitoring that data. If it's something that's really important to you as a sponsor, and maybe you found out that you want to modify an outcome measure because there's something that's new come up in the literature from other people investigating the same indication, then you would need to go back and modify your monitoring plan to make sure that you don't find yourself in a situation at the end of your natural history study that you don't have a good control group if you want to end up using that data in the future. So I think I would treat it the same as an interventional database. If you're adding in data, you just need to make decisions on where your cutoff points are and what your instruction is to the site.
Yeah, you're going to really need to consider the impact on those changes on the subjects that are in the database and then the new subjects. Obviously, with the new subjects coming in, those changes will be easier, and you'll just be entering any of that new data. But when you modify an existing field, you may be removing data that you wanted based off the setup of that in the database. So it's really, you need to consider what those changes will be and how they will impact the new subjects and how they'll impact the existing subjects that are in the database. Because then there would be rework for the sites and for clinical and everybody if data changes. And there's ways working with database programmers to make sure that they know how that data what will happen with the data. And our database programming team is pretty astute in helping us to figure out what's going to happen to that data when we make even the slightest change to that database.
Yep. Yeah, I think it's a great point to consider how these changes will not only impact the data collection prospectively, but also, what does that do to the data that you've already collected for the existing patients? And another thing that you said that also really resonated is, this idea that I think we take for granted that sometimes we assume that sometimes changes are instantaneous, but really it's an iterative and a collaborative process between clinical, the programmers, and to understand that any change, whether big or small, requires a full impact analysis as well as potentially some downtime. So
yeah, definitely.
Yeah. I think it just keeps coming back to risk and making sure that you're taking an integrated risk approach where you're having those cross-functional conversations and having, an understanding of the impacts across the board when you make these changes.
Yeah, thank you so much. So we're at the top of the hour, and so I wanted to I wanted to thank all of our attendees for joining us today. We hope that you found this webinar informative, and please feel free to reach out if you have any further questions. We'd love to hear more from the rest of you about any feedback that you have. And like I mentioned before, please keep an eye out on the invitation for the third chapter of our webinar series, which will come winter of this year. So thank you so much, everyone. Have a good afternoon.
Thank you, everyone.
Thank you so much.
Thanks.
Thank you.
Bye.